Welcome back to the Deep Dive. Today we're stripping everything down to the absolute basics. And I don't mean basics as in easy. I mean basics as in the fundamental machinery that allows you to exist, to think, and to move. We are getting very physical today.
It's honestly the perfect time to do it. We often get so lost in the abstract stuff, psychology, sociology, economics. But underneath all of that is this biological machine operating with just ruthless efficiency.
Exactly. And I want to start with a scenario that I think everyone can relate to. So picture this. You're at the gym, or maybe you're just running down the street to catch a bus that's pulling away. You're pushing your body to its limit.
Right. We all know that sensation. Your chest feels like it's going to explode.
Your heart is just hammering against your ribs. You're sweating like crazy.
That right there is the sensation of a system under absolute siege.
Right. But if you look at the notes for today, what's actually happening under the hood in that moment is incredible. It is a logistical nightmare.
Oh, complete.
My muscles are screaming for energy, which means they need oxygen. So my lungs have to ramp up their respiration rates immediately. My heart has to beat faster to pump that oxygenated blood out to the tissues.
And it's not just the delivery system that's working overtime. Your blood vessels actually have to dilate to accommodate that massive flow. Your kidneys are suddenly adjusting their filtration rates to handle all the metabolic waste being dumped into the bloodstream. Every single system is spiking.
But then, and this is the part that always feels like magic to me, you catch the bus, or you finish the set and drop the weights, and almost immediately the chaos just starts to recede.
The heart slows down.
The breathing steadies, the sweat dries up.
And that return to baseline is essentially the holy grail of biology. We call it homeostasis.
Homeostasis. It's one of those words we hear in high school biology and then immediately forget. But it's essentially the body's prime directive, isn't it? It's the maintenance of a stable internal environment despite the absolute chaos happening outside.
Precisely. Because whether it's 100 degrees outside or 10 degrees, your internal temperature stays exactly the same. Whether you eat a giant bag of sugar or you fast for an entire day, your blood glucose stays within a very specific, narrow range.
Right.
But here is the catch. No single organ can do this alone.
It takes a village, or in this case, a whole body. And that brings us to the concept of coordination.
Right. Coordination is the process where two or more organs interact and complement each other's functions. Just like you noted with the gym example, the heart needs the lungs need the diaphragm needs the muscles.
They all have to talk to each other.
Exactly. And to manage this massively complex orchestra, the body relies on two major command systems.
The first one being the endocrine system, right?
Yeah.
That's the chemical postal service. It uses hormones to integrate activities.
Yes. It is highly effective, but it's generally a slow burn. It's meant for long-term regulation, so things like growth, metabolism, reproduction.
But if you accidentally touch a hot stove, you can't exactly wait for a hormone to circulate through your bloodstream to tell you to move your hand.
No, you'd be severely burned. You need speed. You need a fiber optic network.
And that is our mission for this deep dive. We are exploring the neural system, the command and control center.
This is the system that provides an organized network of point-to-point connections for quick coordination. And when we say quick, we are talking milliseconds.
So here is the roadmap for today. We are going to dissect this system layer by layer. We will start with the hardware, the neurons. Then we are going to look at the spark, the actual electricity, the action potential.
That's the really fun part.
It is. After that, we'll map out the network architecture, the central and peripheral systems, and finally, we will look at the input devices, the eyes and ears that actually feed data into this machine.
It is a wild journey. You're going from the microscopic movement of a single ion across a membrane all the way up to the conscious experience of seeing the world.
Let's start at the bottom then. The fundamental unit, the neuron. I feel like we have all seen that cartoon drawing of a neuron in textbooks. It looks like a splattered egg with a really long tail.
That is a fair visualization, honestly, though the reality is far more complex. The neuron is a microscopic structure that is specialized for one specific thing: detecting, receiving, and transmitting stimuli.
So let's break down that anatomy. We have the cell body. This is the headquarters of the cell.
Yes, it contains the cytoplasm and all the typical organelles you would find in any regular cell: mitochondria, a nucleus, all of that.
It's like a brand of breakfast cereal. But I know they are granular bodies found in the cytoplasm. What is their actual function?
They are essentially clusters of rough endoplasmic reticulum and free ribosomes. So if you remember your cell biology, that means they are the protein synthesis factories of the neuron,
Okay.
and you find them inside the cell body and also out in the dendrites.
Speaking of dendrites, these are the short fibers branching out of the cell body. They look exactly like the roots of a tree.
Or antenna, because that is their function. They branch repeatedly, projecting out of the cell body. Their entire job is to catch the incoming signal and transmit those impulses towards the cell body.
They're the receivers.
Exactly.
Okay, so the signal comes in through the dendrites, hits the cell body, and then what? Has to go somewhere.
It heads straight to the axon. The axon is that long fiber you mentioned. It's the transmission line. The distal end, or the far end of the axon, is highly branched, and each of those terminal branches ends in a bulb-like structure called a synaptic knob.
The synaptic knob. This is essentially the loading dock.
That is a brilliant way to put it. Inside that knob, you have these synaptic vesicles. Think of them as little cargo ships containing chemicals called neurotransmitters.
Right.
So the axon's job is to transmit nerve impulses away from the cell body down to a synapse, or a neuromuscular junction.
Okay, so just to trace the flow of information, dendrites pull it in, the cell body processes it, and the axon sends it out.
Correct. Now, nature loves variety, so not all neurons look exactly the same. We classify them based on the number of axons and dendrites they have.
Right, I've got the list from the source material right here. We have multipolar, bipolar, and unipolar. And multipolar seems to be the standard model we always talk about.
Multipolar neurons have one axon and two or more dendrites. This is the most common type by far, and it's what you find in the cerebral cortex, the thinking part of your brain.
And then bipolar.
One axon and one dendrite. It's a very simple input-output structure. You find these in very specific sensory areas like the retina of the eye.
And unipolar.
The cell body has one axon only, 0 dendrites. These are usually found in the embryonic stage. As the nervous system develops, they typically differentiate and become more complex.
Got it. Now, we really need to talk about insulation, because I know from wiring my own house that you cannot leave copper wires exposed or they short out against each other. The body has a very similar problem with all these electrical fibers, right?
It absolutely does. And the biological solution to that is myelination. We essentially have two types of axons in the body, myelinated and unmyelinated.
And in the myelinated ones, we have these special cells called Schwann cells. They wrap themselves around the axon like a bandage.
Like a bandage or like thick electrical tape, they form what we call the myelin sheath. But here is the really critical design feature, that wrapping isn't perfectly continuous. There are little gaps.
The nodes of Ranvier.
Yes. If the insulation was perfect and completely continuous, the electrical signal couldn't interact with the outside environment to recharge itself as it travels. These gaps allow the impulse to literally jump from node to node.
Oh, so it's like skipping a stone across the water versus trying to drag it through the mud.
That is the perfect analogy. This jumping process is called saltatory conduction, and it makes the transmission incredibly fast. You find these myelinated fibers in spinal and cranial nerves where speed is literally a matter of life or death.
And the unmyelinated fibers.
They actually still have a Schwann cell enclosing them, but it doesn't form that thick, multi-layered myelin sheath. So the electrical transmission is a lot slower. You see these mostly in the autonomous and somatic neural systems.
Managing things that just don't require split-second reflexes, like digestion.
Exactly.
Okay, so we built the cable, we have the input, the output, and the insulation. But a cable is completely useless without a spark. And this is where the biology gets really heavy really fast. We need to talk about the nerve impulse.
The spark itself.
To understand this, we have to understand that a neuron is an excitable cell. And it is excitable because its membrane is polarized.
Polarized is the key word here. Even when a neuron is doing absolutely nothing, when it is just in its resting state, it is under massive tension. It is like a drawn bowstring or a fully charged battery.
Let's look at the chemistry of that battery.
Yeah.
Because it all comes down to ions. Inside the axon, which is called the axoplasm, and outside the axon, the fluids are completely different.
Very different. Inside the axon, you have a high concentration of potassium ions, that's K+, and you also have a lot of negatively charged proteins. But you have a very low concentration of sodium ions, Na+.
Okay, so let me lock that in. Inside equals high potassium, low sodium, and a bunch of negative proteins.
Right. And outside the axon is the exact inverse. You have a low concentration of potassium and a super high concentration of sodium.
Now, normally nature hates gradients. If I spray perfume in the corner of a room, it immediately spreads out evenly. Why doesn't the sodium just rush in and the potassium rush out to even things up?
Because the neuronal membrane is a very strict gatekeeper. It is selectively permeable. In this resting state, the membrane is somewhat permeable to potassium. It lets a little bit of it leak out, but it is nearly impermeable to sodium. It blocks the sodium almost completely.
And what about those negative proteins inside?
They are simply too big to cross the membrane. They are trapped inside.
But there is something else actively keeping this imbalance alive. A biological machine. The sodium-potassium pump. This isn't just a passive gate.
No, this is an active transport mechanism. It requires actual energy, ATP, to run. Think of it like a bouncer at a busy club who is actively grabbing people and throwing them out the door. This pump physically transports 3 sodium ions outwards for every 2 potassium ions it drags back into the cell.
Let's do the math on that. 3 positive charges get thrown out. 2 positive charges get pulled in. So every single time that pump cycles, the inside of the cell loses a net positive charge.
Correct. And it is doing this constantly. So over time, combined with those trapped negative proteins we talked about, this creates a situation where the outer surface of the membrane becomes positively charged and the inner surface becomes negatively charged.
And that difference in charge across the membrane is the voltage.
That is the resting potential. The cell is sitting at a negative voltage relative to the outside world, just waiting, ready to fire.
Okay, so the gun is loaded, the battery is fully charged. Now, a stimulus hits, a touch on the skin, a sound wave, or a signal from another neuron. What actually pulls the trigger?
The action potential. Let's look at one specific spot on the axon. We'll call it site A. When an adequate stimulus hits, the membrane properties at site A change instantly. The membrane suddenly becomes freely permeable to sodium.
The bouncer just steps aside and opens the velvet rope.
Completely. And because you have that massive concentration of sodium outside, just desperate to get in, and because the inside is negative, which magnetically attracts the positive sodium, the sodium just crashes in.
A total flood.
A rapid influx. And this massive influx of positive ions reverses the polarity at that specific site. Suddenly, the outer surface becomes negative and the inner surface becomes positive. This event is called depolarization.
It flips from negative to positive, and that massive swing is the electrical signal itself.
Yes. That electrical potential difference is the action potential. That is your nerve impulse.
But a signal isn't very useful if it just stays at site A. It has to travel all the way down to site B and C and D to reach the end of the nerve. How does it physically move?
Imagine the stadium wave. At site A, we are now positive on the inside. But right next door at site B, the membrane is still at rest. It's still negative inside and positive outside.
So we had a highly positive patch right next to a highly negative patch.
And basic physics takes over. An electrical current flows on the inner surface from the positive site A to the negative site B. Meanwhile, on the outer surface, the current flows from the positive site B back to the negative site A. This completely closes the circuit.
And that flow of current acts as the stimulus for site B to open its own floodgate.
Exactly. The polarity at site B reverses. The action potential effectively moves from A to B. And this exact domino effect races down the entire length of the axon in a fraction of a second.
But we have a huge problem at site A now. It is full of sodium. It's fully depolarized. It cannot fire again until it resets.
Right. And that reset is the repolarization phase. The membrane's permeability to sodium is extremely short-lived. Those gates shut off almost as fast as they opened. And immediately after they shut, the permeability to potassium suddenly rises.
So the sodium gates slam close, and the potassium gates swing wide open.
And since there's still a high concentration of potassium inside, and the inside is now positive, the potassium just diffuses rapidly outside the membrane. Since potassium is positive, its massive exit restores the negative charge on the inside of the cell.
And the battery is officially recharged.
The resting potential is restored. The fiber is ready for the next stimulation.
It is honestly incredible to think that every single thought I have, every memory, every muscle movement, every word we are saying right now is just this microscopic sodium-potassium dance happening billions of times a second.
It's the currency of consciousness.
So the signal travels down the axon as this electrical wave. It finally reaches the end of the line, the synaptic knob. But here's the complication. Neurons don't actually physically touch each other. There's a gap.
The synapse. It's the junction between the presynaptic neuron, the one sending the signal, and the postsynaptic neuron, the one receiving it. The physical gap itself is called the synaptic cleft.
So how does this signal jump the canyon? Because electricity can't just spark across empty space like that inside the body.
Well, there are 2 ways. First, and this is quite rare, we have electrical synapses. Here, the membranes are in such close proximity, almost physically touching, that the electrical current can actually flow directly through gap junctions from one neuron to the other.
That sounds a lot faster.
It is significantly faster, but it's very rare in the human neural system.
Why is that? If speed is always good in a biological system, why isn't everything wired electrically?
Because electrical synapses are very rigid. They are an all-or-nothing transmission. They don't allow for much nuance or control. And that is why the vast majority of our nervous system relies on chemical synapses.
Right, chemical synapses allow for modulation. So walk me through that chemical crossing.
When the action potential reaches the axon terminal, it stimulates those synaptic vesicles we mentioned earlier, the little cargo ships. They move right up to the plasma membrane, fuse with it, and basically burst open.
Releasing all those neurotransmitters directly into the cleft.
Exactly. The chemicals float across that tiny gap, and on the other side, the postsynaptic neuron has highly specific receptors. The neurotransmitters bind to these receptors just like a key finding its exact lock.
And turning that key does what?
It opens specific ion channels on the receiving neuron. Ions rush in, and a brand new electrical potential is generated on the other side.
And I assume this is why chemical is so much better than electrical, that new potential isn't always just telling the next cell to fire, is it?
No, it's not. The new potential can be excitatory, meaning it tells the next neuron to fire its own action potential, or it can be inhibitory. It can actively tell the next neuron, do not fire, stay quiet.
Which is how the brain filters out background noise.
Precisely. It allows for complex processing, decision-making, prioritization. If every single signal was just stuck in the on position, your brain would be in a constant state of seizure. You need the brakes just as much as the gas pedal.
So we have the hardware, the neuron, we have the spark, the action potential, and we have the connection, the synapse. Now let's zoom out a bit and look at the actual network architecture. The nervous system as a whole is divided into two main commands, the CNS and the PNS.
Headquarters and field operations. The central neural system, or CNS, is just the brain and the spinal cord. This is the site of all information processing and control.
And the PNS.
The peripheral neural system. These are all the nerves associated with the CNS that extend outward to the rest of the body. The PNS is essentially the massive bundle of wires connecting headquarters to the outside world.
And the PNS has two specific types of fibers based on the direction the signal is going, right?
Afferent and efferent fibers.
I always use a simple mnemonic for this. Afferent starts with A for arriving.
That's a good one. Afferent fibers transmit impulses from the tissues and organs to the CNS. They bring the sensory intel into headquarters.
And efferent starts with E for exiting.
Correct. Efferent fibers transmit regulatory impulses from the CNS out to the peripheral tissues. They carry the outgoing orders.
Now, zooming in on the PNS, we have further subdivisions based on exactly what is being controlled. First, we have the somatic neural system.
The somatic system relays impulses from the CNS straight to your skeletal muscles. This is your voluntary control. You consciously decide to pick up a coffee cup, your somatic system executes that command.
And then we have the autonomic neural system.
This one transmits impulses from the CNS to the involuntary organs and smooth muscles. Your heart rate, your digestion, your sweat glands, the stuff that runs purely in the background without you having to think about it.
And the autonomic system is split yet again into that famous duo, sympathetic and parasympathetic.
The accelerator and the brake. The sympathetic system is your fight or flight response. It preps the entire body for a crisis. The parasympathetic is rest and digest. It conserves energy and handles maintenance.
I see a note in the source material here about the visceral nervous system, too.
Yeah, that's effectively the part of the peripheral system, the whole complex of nerves, ganglia, and plexuses that specifically travels to and from the viscera, your internal organs. It's the dedicated communication line for your gut.
Okay, let's head up to headquarters. The command center, the human brain.
Truly the most complex structure in the known universe.
And because it is so vital, it is heavily armored. Obviously, it's inside the skull, but it's also wrapped in these layers called the cranial meninges.
Right, it's an internal crash helmet. There are 3 layers. The outer layer is the dura mater, which literally translates to tough mother in Latin.
Tough mother, that's great.
It is very durable and thick. Below that is the middle layer, the arachnoid. It's very thin and web-like. And the innermost layer is the pia mater, which is highly delicate and is actually in direct, intimate contact with the brain tissue itself.
Dura, arachnoid, pia. Now, let's essentially dissect the brain itself. We generally divide it into the forebrain, midbrain, and hindbrain. Let's start with the forebrain. This is the main processor.
Right. It consists of the cerebrum, the thalamus, and the hypothalamus. The cerebrum is the massive bulk of the brain. It's divided longitudinally into the left and right cerebral hemispheres.
Which are connected by the corpus callosum.
Yes, a massive tract of nerve fibers that acts as a bridge, letting the two sides communicate instantly. The outer surface of the cerebrum is called the cerebral cortex. This is what we refer to as the gray matter.
And why exactly is it gray?
Because it is absolutely packed with neuron cell bodies. It looks grayish in a physical cross-section. The inner part of the cerebrum is called the white matter because it's full of those myelinated axons we discussed earlier. The white color comes directly from the fatty myelin sheaths.
And the cortex is really where the magic happens. It has motor areas, sensory areas, and these large regions called association areas.
The association areas are crucial to who we are. They aren't clearly sensory or clearly motor in function. Instead, they are responsible for the highly complex functions like memory, communication, and intersensory association. It's where you put all the pieces together to understand context.
Buried below the cerebrum, we have the thalamus.
The thalamus is essentially the grand switchboard. It's a major coordinating center for sensory and motor signaling. Almost all sensory info from the body goes right through the thalamus before being routed to the cortex.
And right at the base of the thalamus lies the hypothalamus.
The master controller of your baseline drives. It regulates body temperature, hunger, thirst. But incredibly, it also contains neurosecretory cells that secrete hypothalamic hormones. So it acts as the primary interface between the nervous system and the endocrine system.
And we absolutely cannot skip the limbic system.
No, we cannot. The inner parts of the cerebral hemispheres, along with deep structures like the amygdala and hippocampus, form the limbic system.
Often called the lizard brain.
Right, the emotional brain.
Yeah.
It regulates sexual behavior, motivation, and intense emotional reactions. Excitement, profound pleasure, uncontrollable rage, primal fear. The hippocampus, which is part of this, is essential for converting short-term memory into long-term memory, which is exactly why smells, which feed directly into this limbic system, can trigger such overwhelmingly strong emotional memories.
Moving down the stalk, we hit the midbrain.
It's located right between the thalamus of the forebrain and the pons of the hindbrain. A key internal landmark is the cerebral aqueduct passing straight through it. On the dorsal side, the backside, you have the corpora quadrigemina. These are 4 round swellings heavily involved in visual and auditory reflexes.
And finally, the hindbrain, the ancient life support system.
Comprising the pons, cerebellum, and medulla oblongata. The pons mostly consists of heavy fiber tracts that interconnect different major regions of the brain.
Then the cerebellum. And that word means little brain.
It does. It has a very convoluted, folded surface in order to pack in as many extra neurons as possible. It's entirely responsible for coordination, balance, and fine motor skills. When you ride a bike without actively thinking about balance, you are heavily using your cerebellum.
And then there's the medulla.
The medulla connects directly to the spinal cord. It controls the absolute vitals: respiration, cardiovascular reflexes, gastric secretions. You can damage parts of your cortex and lose your personality or memory, but if you severely damage the medulla, the lights just go out. It runs the core life support.
Now, sometimes even the brain is just too slow. If I accidentally touch a hot stove, I need to move right now. I cannot wait for the afferent nerve to tell the thalamus to forward the memo up to the cortex to have an association area meeting about whether it hurts or not.
No, you would lose the hand. That is exactly why we evolved the reflex action. It's an entirely involuntary response to peripheral stimulation that occurs without any conscious thought whatsoever.
It uses something called the reflex arc. Let's walk through that hot stove example step by step.
Okay. Step 1, a receptor in your skin detects the extreme heat. Step 2, an afferent neuron immediately transmits that impulse via the dorsal nerve root straight into the CNS, but specifically into the spinal cord.
It just stops at the spine.
For the immediate decision, yes. Step 3, the processing happens directly in the spinal cord. Step 4, an efferent neuron instantly carries the motor signal away. Step 5, the effector, which is the muscle in your arm, contracts.
And I pull my hand away?
And you do it before you even feel the pain. The actual pain signal continues traveling up to the brain on a separate track and arrives milliseconds later. That is why you physically pull back first, and then you process the pain and yell out.
It's just an incredible override system. Okay, so we've covered the processor and all the wiring. But how does the data actually get in from the outside world? We need to talk about the senses, specifically the transducers, the eye and the ear.
Transducer is the exact right word to use here. They are biological machines that convert physical energy in the environment, like light photons or sound pressure waves, into electrical nerve impulses.
Let's start with vision, the eye. Let's do the anatomy first. It's a sphere built with three distinct layers.
The tough external layer is the sclera, which is dense connective tissue. The front portion of this layer is the cornea. That's the clear window you look through. The middle layer is the choroid. It contains lots of blood vessels and looks bluish. In the front of the eye, it thickens to form the ciliary body.
And the ciliary body is what holds the lens in place.
Yes, the ciliary body holds the lens and also extends forward to form the iris, which is the pigmented, visible, colored part of your eye.
And the pupil is essentially just the physical hole in the middle of the iris.
A heavily regulated hole. The muscle fibers of the iris actively adjust the pupil's diameter to constantly control how much light gets in.
Okay, and the inner layer is the retina. This is the photographic film in the back of the camera.
Right. The retina contains three separate layers of neural cells. Going from the inside out towards the back, you have ganglion cells, bipolar cells, and finally the photoreceptor cells.
The photoreceptors being the absolute stars of the show here. We have rods and cones.
Right. Rods handle scotopic vision, which is twilight or night vision. They are incredibly sensitive to even tiny amounts of light, but they do not detect color. They contain a purplish-red protein called rhodopsin, or visual purple.
And rhodopsin contains a derivative of vitamin A, correct? Which is why my mother always told me carrots are good for my eyes.
She was absolutely right. A severe vitamin A deficiency causes night blindness because you can't synthesize enough rhodopsin. Now, the cones are for photopic vision, daylight, and color vision. We have three specific types of cones responding to red, green, and blue light. When all three are stimulated equally, our brain synthesizes that as the sensation of white light.
I see a note in the source outline here comparing the blind spot to the fovea.
The blind spot is exactly what it sounds like. It's the physical location where the optic nerve leaves the eye. Because the nerve is there, there is no room for photoreceptor cells. If an image focuses precisely on that spot, you simply see nothing.
Right.
But slightly lateral to the blind spot is a yellowish pigmented area called the macula lutea. And right in its center is a tiny pit called the fovea.
The fovea is essentially the high-resolution center of the eye.
It is packed incredibly densely with only cones. It is the point of greatest visual acuity or resolution in your entire field of vision. When you read a book or thread a very fine needle, you are pointing your fovea directly at it.
Now, explain the actual chemical mechanism. How does light hitting the back of the eye become a pulse of electricity? Because this is the real chemical aha moment for me.
It is honestly beautiful chemistry. The photosensitive compounds, the photopigments in our eyes, are composed of opsin, which is a protein, and retinol, which is an aldehyde of vitamin A.
So the opsin and retinol molecules are physically stuck together?
When it's dark, yes. But when a photon of light hits the retina, it physically induces the dissociation of the retinol from the opsin. It literally knocks them apart.
And that separation changes the physical shape of the opsin protein.
Yes, the 3D structure changes. And that structural change immediately alters the membrane permeability of the photoreceptor cell.
And we know from earlier what changing membrane permeability does. It allows ions to move, which creates a potential difference.
Exactly. This generates an action potential in the photoreceptor, which travels down to the bipolar cells, then to the ganglion cells, and finally generates full action potentials in the optic nerves, carrying the signals straight to the visual cortex.
So my entire sense of vision is quite literally just the physical breaking of a chemical bond by a photon, which is then translated into a spark of electricity.
That is exactly what seeing is at a molecular level.
That is just mind-blowing. Okay, let's look at the final mechanism for today, the ear. Hearing and balance.
Right. The ear is anatomically divided into the outer, middle, and inner sections. The outer ear is the pinneter, the floppy cartilage part you can actually see, and the external auditory meatus, or the ear canal. Their job is just to collect and funnel sound waves right to the tympanic membrane, the eardrum.
Then we cross into the middle ear, and this is where we find the three smallest bones of the human body.
The ossicles. They are the malleus, incisius, and stapes, or hammer, anvil, and stirrup. They are attached to each other in a chain and act as a mechanical lever system. They amplify the incredibly tiny vibrations from the eardrum and transmit them with force to the oval window of the cochlea.
And force is needed because the inner ear is completely fluid-filled. It's much harder to move fluid than air.
Exactly. The cochlea is the coiled portion of the inner ear labyrinth. It looks exactly like a snail shell, and it's divided internally into fluid-filled chambers. the scala vestibuli, scala media, and scala tympani.
So the stapes bone punches against the oval window, and that physical push creates a wave in the fluid inside the cochlea.
It creates pressure waves in the lymph fluids, and these fluid waves induce a physical ripple in a very specific internal membrane called the basilar membrane.
And sitting right on top of that basilar membrane is the organ of Corti.
The actual biological microphone of the body. The organ of Corti contains thousands of specialized hair cells acting as auditory receptors. As the basilar membrane ripples up and down, these hair cells are pushed and bent against a rigid, thin, elastic membrane sitting just above them called the tectorial membrane.
So the mechanical shearing force physically bends the tiny hairs.
And just like the eye, that physical bending forces ion channels at the base of the cell to open. Nerve impulses are instantly generated, sent via afferent neurons to the auditory nerve, and then routed to the brain's auditory cortex.
Amazing. Now finally there's balance, the vestibular apparatus. And this is completely separate from the hearing mechanism in the cochlea, right?
It is structurally attached, located just above the cochlea, but it serves a totally different function. It has 3 semicircular canals arranged perfectly at right angles to each other. So basically mapping the X, Y, and Z axes of 3D space.
Which allows them to detect any rotation of the head?
Yes. The base of each canal is swollen in an area called the ampulla, and inside is a structure called the crista ampullaris, which has more of those sensory hair cells. When you spin your head, the fluid inside the canals lags behind due to inertia, physically bending the hairs and sending a signal.
And there's also the otolith organ.
Right. The otolid consists of the saccule and utricle. They contain a projecting sensory ridge called a macula. While the canals handle rotation, the macula detects basic gravity and linear acceleration.
So the semicircular canals tell me if I'm turning my head, but the otolith tells me if I'm suddenly going up in a fast elevator.
Precisely. It allows you to maintain body balance and posture against gravity without ever actively calculating it.
When you step back and look at this entire system we just mapped out, it is almost overwhelming.
It really is the ultimate biological integration. The ear literally converts mechanical air pressure into a fluid wave, into an electrical spark. The eye converts incoming photon into a chemical shape change, then into electricity. The neuron constantly acts as a mechanical pump just to maintain a battery charge so it can fire. And the brain synthesizes billions of these inputs simultaneously to keep you alive.
And I think that actually leads us perfectly to the big question,
Yeah.
the provocative thought we promised at the beginning of the show.
Oh, right. So we have spent this entire hour meticulously breaking down the sheer mechanics of it all. We now know that vision is essentially just a molecule of retinal separating from opsin. We know that a thought traveling down a nerve is just sodium rushing through a protein gate into an axon.
Right. It's just highly organized physics and chemistry.
So here is the real question for the listener. If the entire neural system is merely a massive series of ion gradients and chemical switches, just sodium in, potassium out over and over, at what precise point does a basic chemical reaction become a conscious thought? At what exact point does a firing chemical synapse become the subjective feeling of love, or a vivid memory of your grandmother? Where, in this incredibly complex wet web of axons, dendrites, and pumps, does you actually exist?
That is both deeply terrifying and incredibly beautiful. It's definitely something to ponder while your sodium-potassium pumps quietly keep working in the background. Thank you for joining us on this deep dive into neural control and coordination. Stay curious.